Right, so if you look at the last video where we just talked about the microscope, the light microscope, and there are two types of experiment that you've probably seen or done in school. One is looking at a cheek cell of an animal, and the other one is looking at a plant cell, probably an onion cell. And what you might have to do or need to do is just be able to describe what happened. So fortunately we've got a lot of similarities, so let's have a look at those first. So we have let's start off with the glass slide. So in both diagrams there is a glass slide. We'll do the animals, the cheek cell first. So the glass slide is the bit that we put everything on. So there's the glass slide there, and then we have a mounted needle. So have a look and say, which do you think is the mounted needle? So the mounted needle is the pointy thing there, useful for poking things and moving things around. And then lastly, both have a cover slip. So the cover slip is the thing we put over the whatever it is we're trying to magnify, so that cover slip is there. And you can see it's the same for both of them. So you should be able to label those three, doesn't matter what, whether you're looking at a cheek cell or an onion cell. So let's have a look at the animal cell first of all, the cheek cell. So first thing we need to do, if you're doing this experiment, we would use a cotton bud and you'd rub it inside your mouth, on your cheek, inside your mouth, to pick up some cells. Okay, what you would do then is you would put something called methylene blue, probably because it's blue and it's methylene, onto the glass slide. You could then take your cotton bud. Now that's not a cotton bud, but it does say cotton bud. Let's imagine it's a cotton bud, and you'd rub it in the methylene blue, and that would get some of the cheek cells in the methylene blue, so we can then look at them under the microscope. Now, something else worth knowing, we call this biological staining. So the methylene blue will stain those cheek cells and it will make them stand out really nicely when we look at them in the microscope. So biological staining is what that is. There we go. So we are, methylene blue. And we put the methylene blue on the glass slide. We rub the cotton bud in the methylene blue, then we use the mounted needle to lower the cover slip into place, and then we can use the light microscope to examine it. Always worth, if you have to describe it, always worth mentioning, use the microscope to examine it, because you'll probably get some marks for that. Right, so let's have a look at the onion cell. So again, cover slip. Now some people get cover slip and glass slide mixed up. The glass slide is always the big bit at the bottom. Okay, and then we have our mounted needle again, except this time what we do is we have, instead of methylene blue, we have a blob of iodine solution. And again, biological staining. So the iodine solution helps the onion cells stand out nicely when we magnify them. And we use forceps to pick up. So the forceps, a bit like my fingers here, my finger and my thumb, they allow you to pick up something. So we can pick up part of the onion, pop it onto the slide, put some iodine solution onto that, and then put the cover slip on top with the mounted needle, just as we did before. Okay, sorry this video is a bit longer. Hopefully you managed to make it to the end, and thank you very much for listening, and we'll see you in the next video. Thank you very much.